All right, scholars, I'm going to take a few moments to give you a video on mutations. Since I won't be here on Tuesday or Thursday, I thought this would prove very helpful for you to explain this and get your minds around it. You've probably seen some of this already in biology in the past, but this is a bit of a refresher on the idea of mutations. All a mutation is a change in DNA sequence. Sometimes it's significant, sometimes it's not. For instance, here's a sample DNA sequence. I'm going to use this throughout our lesson today. Mutations affect things by changing the DNA. If we were to transcribe and translate this, let's say we created the RNA for this. Using Chargaff's rule, we would have, I'll use a different color just to make this visually distinct. We would have A, we would have U, we would have G, then we'd have G, C, U, we'd have A, U, then we have U, A. That's the transcription process, making messenger RNA. Now, assuming this has been properly set up and been, let's say, edited, introns removed, all that information, it gets out to the ribosome that's used to make your protein. Sorry, guys, forgot the word. Protein. Your protein would be based on groups of 3 letters. Each group of 3 letters, remember, is called a codon. So, using the genetic code charts we've used before, AUG is methionine, GCU, which I have one sitting over here on my computer to make it easy, GCU would be alanine, AAU would be ASN, and the 4th one, UUA, would be LEU. Alright, so that's if everything goes normally. What a mutation does is it changes the DNA, which changes the RNA, which changes the protein. We can have what we call little mutations that we call those point mutations. Remember, a point mutation is just what it sounds like. It's a mutation at one point. One point in this process. So for instance, let me give you an example of a point mutation. What if, instead of this being AAC, it was AAA? Well, that would mean that this now is UUU. Instead of UUU, remember, if you look at your chart, actually is not leucine, it becomes phenylalanine. So, we've changed one letter at one point, creates a different RNA, thus creates a different protein. That is called a point mutation. It affects it in one place. It's a substitution. It's also called a substitution mutation. We substituted one base for another. That's all we did there. Changed one base, changed it for another base. That's one type of mutation you can have as a point mutation. Another type you can have would be a deletion or an insertion. Allow me to show you what that would look like. Let's go back to how this originally looked. Let me go back to my default. It originally looked like this. These back as they were. I had this, forgive me, UUA, so that means it's AAC, so it's UUG. All right. So let's say we have that as our base process. We already looked at the substitution. Let's look at what a deletion would look like. A deletion, what it does is it removes a letter. For instance, let's say I took out this right there. Well, here's how that affects things, guys. If this isn't there, that means this letter is not going to be there in the mRNA. The whole process of making protein has to work in groups of 3. It has to work in codons. So where I've drawn these lines before, you have to change them. GC, now be GC, A, U, U, and I guess these last two aren't going to get used. What that means is this is likely going to be different. GCA now, GCA according to our genetic code is alanine. Oh, look, that one stayed the same. But AUU, when you go and change it, would now be isoleucine. And UG, well, you have to have 3 letters to code for anything, so that's gone. This is a deletion. We also could call a deletion a frame shift mutation, okay? This is called a frame shift. Because what we've done is, if you think of each one of these boxes, each one of the codon boxes as a frame of reference, a reading frame perhaps, we have shifted them all because we took a letter out. We changed everything after this by changing just one thing right there. And it affected things down the line without doing a lot of damage right here. It's going to affect everything else along the way. The opposite of a deletion would be an insertion. So an insertion would also be a frame shift mutation. Thing is, instead of taking out a letter, we're adding a letter in. That's going to shift all the groups of 3, and we're going to end up with change all the way down the line. Every single one of them down the line. So let's say, for instance, here we originally had, let's see, forgive me for this. We originally had this at GCA, I'm going to put it back to the way it was for us. Okay, back to how it was. Well, if this was the original grouping, like this, if I were to insert a letter, maybe I decide to throw in, oh, I don't know, another A right here. Now all my frames have to shift again. This is right here, that will be right there, and that will be right there. Let's see what that does to the proteins. AAA codes for lysine. UUU, triple uracil, codes for phenylalanine. It's all right there. That's how this process works. Lysine, phenylalanine. That is the process that goes. We've inserted, we changed everything. This is also a frame shift. So these are both frame shift mutations. This is our first video on this. I intend to put more videos out for you in the second part of this, which I'll record in just a moment.